Hello everybody, welcome to our live Phenomics approach on plant breeding. My name is Roberta and I coordinate the scientific committee of GVANC. Before we start with the lectures, I'm going to make a brief presentation about GVANC and some of the activities that our group have been developing over the last few years. GVANC is short for Genetics and Plant Breeding Group, Professor Roland Vankovskis. And this name was a tribute to Professor Roland for his huge contribution to many areas, especially in quantitative genetics, population genetics, and conservation of genetics resources. We are formed mostly by graduate students, but we have a few undergraduate members as well from Luiz de Queiroz College of Agriculture, campus of University of São Paulo, who are interested in genetics and plant breeding. And the group's mission is to integrate academies, professors, and professionals focusing on preparing future genetics and breeders. These are the current members. We have a general coordination, which is formed by Professor Baldin Pinheiro and the grad students Mayara de Oliveira and Jessica Ferraresi, and also 5 other committees that are logistics, communication, secretary, scientific, and treasury, each of them with their specific roles in the group. Among our activities, the main one is holding the International Meeting on Plant Breeding, which is part of the Plant Science Symposia series, a series of scientific conferences sponsored by Corteva AgriScience. In fact, it was the organization of this event that was the main motivation for the group's creation in 2017. And so far, we have held 3 editions of the event. The first two editions took a more general approach on several techniques applied to plant breeding. And the third one focused on artificial intelligence and machine learning. During the international meeting, in addition to the lectures, GVANC also offers mini courses that are given by some of the participating speakers. These 2 first photos are from the 2 first event, and last year in the 3rd edition, we offered 2 courses during the international meeting. This year, the international meeting will take place between September 30 and October 2nd, and will bring the theme Gene Editing by the name Uncovering the Power and the Secrets Behind DNA Modifications. The event will be online and open access, so I invited you to follow GVANC's social media that more information will be released soon. Last year, we also organized a session during the 10th Brazilian Congress of Plant Breeding, which was called Smart Tools for Plant Breeding, and had the participation of international research speakers. GVANC has a YouTube channel where you can find lectures with some of the speakers that participate from our event, as well as almost all the lectures from the past editions of the international meeting. So it really is worth to take a look over there. Talking now a little bit about some of the more common activities of GVANC. We organize courses at the university. Until now, we have already organized 2 of them, and Python course and in software R1. We organize visits to companies and research institutions related to plant breeding as well. Here I put photos of some of them. We have Hideza, Vilmorin, Fibria, Suzano, Yacé Café, and Sakata. And we also held the Topcross and Voice of graduations in the university. The Topcross is an event of lectures that we had already had presentations from students, professors, researchers, and professionals in several teams. And the Voice of Graduates, it's an exchange of experience between former students of the university, which now work actively in genetics and plant breeding. Finally, we enter the Intergen, which is an event held in partnership with other genetics and plant breeding groups from several Brazilian universities, which aims just to promote integration between the groups and the universities. The first edition of the Intergen was made in Lavras in 2018. The second edition occurred last year during the Brazilian Congress of Plant Breeding in Águas de Lindóia, São Paulo. And this year, Intergen happened this week and it was made online. So, like I said before, this was only a brief presentation about our group. I invite you again to follow Givenc's social media. You can find the links in the descriptions below this video, or you can just point your cell phone camera to the QR code on the screen. Also, in the descriptions of the video, you will find an feedback form that we will ask you to answer once you can, because these feedbacks are really important to us to keep improving our events. Now we can start with our schedule. We will have 3 lectures of approximately 30 minutes each. And after the lecture, you have 10 minutes for doing the question. And you can send your question at the YouTube comments that our team will be selecting it and send it to the speaker. Also, between the second and the third lesson, we will have a 10 minutes break. And finally, we will have the panel discussions where the speakers will be discussing the 3 following topics: prospects for use and challenges of phenotyping, entrepreneurship and innovation opportunities in phenomic, and finding a way to enter the phenomics. Now, I would like to call Dr. Annarita Marrano. She has bachelor and master degrees on agriculture biotechnology from University of Bari, PhD degree in biotechnology for the University of Padova, postdoc researchers from University of California, Davis, and now she is interested in identifying the genetics and non-genetics factors that contribute to trait phenotypic diversities in domesticated and wild plant systems. And currently, she works as a researcher at the University of Massachusetts, Boston, where she is studying the genetic control and adaptation to grow in Asian rice. So, welcome, Dr. Annarita. Can you hear us?
Yes. Hello, everybody. Yeah. Thanks to the organizer for inviting me today.
So I'm going to share your screen now on the screen, and you can go ahead with your presentation.
Okay. Thank you, Roberta. So hello, everybody. As I say, I want to thank the organizer for inviting me today, and also all the attendees that are participating to this panel discussion. So today I'm gonna present part of the work that we do at Moyer's lab at University of Massachusetts, Boston. We are interested in understanding drought response and adaptation in rice, and in particular today I'm gonna describe how we studied seed variation in rice grown under drought stress by applying digital tools. So as you may know, rice is among the top 10 crop cultivated worldwide and is a staple food for more than half of the global population. Asia accounts for more than 90% of the rice cultivation, in particular China and India. They lead the rice production worldwide. Since the advent of the Green Revolution, with the identification of dwarf and high-yielding cultivars, rice and in general crop production has increased consistently across the year. However, recently, some predictions are suspecting that in the following years there will be a further increase of the global population to almost 9 million of people. Therefore, as breeders, as plant breeders, we need to further increase yield in many crops, including rice, and we have to do that while not increasing the cultivation land, but also reducing the input consumption of resources. In particular, in the case of rice, we are dealing with a major problem with drought. So in this plot, I'm representing the drought risk that is current occurring in many cultivation area for rice. And as you can see, many of these areas are already facing severe water scarcity. In particular, the area highlighted in yellow have a low medium risk, while the one in red, they have already high drought risk. And as you can see, India and China are already facing are already dealing with drought problems. So in the recent year, rice breeding is trying to, besides increasing yield, but is trying to do that while identifying drought tolerant individuals. And also, this is becoming quite complicated because recent studies has shown that while across the years, rice breeders were selecting for high-yielding and dwarf individuals, at the same time, they were selecting against drought tolerance. For instance, here I'm presenting the work of Ricam et al., where they discovered that one of the major genes responsible for dwarfism and high-yielding in rice, the SAD1, is tightly linked to one of the major QTL controlling drought tolerance in rice. And so they observed that the favorable allele at SD1 was linked to the unfavorable allele for drought tolerance. Therefore, as a conclusion, we can say that most of the rice cultivars that we have today, they are, yes, high yielding, but they are also highly drought sensitive. So in the sense of increasing yield, attention has been devoted to seed weight and size, while yield in rice has three major components, which are the number of panicle per plant, the number of seeds per panicle, but also grain weight. And grain weight, it is strongly and positively correlated with seed size, which is at the same time a very important qualitative trait in rice. So here on the right, I'm presenting a schematic representation of the grain in rice, which is very similar to other cereals. But I want to point out the attention to the Spiker et al., which is the major determinant of seed size and shape, since it's defined the space where the grain will be filled, and so it strongly influences seed size and shape. So due to their strongly correlation to yield, seed size and weight have been extensively studied across the years. So here there is a schematic representation of the major genes and QTL that have been identified by many research groups. And as you can see, they are involved in many metabolic pathways. But most of them, they intervene in the two major phases of grain development, which is cell proliferation and cell expansion. However, while many studies have been focused on understanding the genetic control of seed size and weight in rice, few of them are applied are trying to understand which is the drought effect on these traits, and in particular, if there is a different genetic control under drought condition. So our study has as main objective the main objective trying to understand which is the genetic control of these traits under limited water condition, and how we are doing this. We are studying seed variation in global multi-parent advanced generation intercross population grown under drought and well water condition. But we also understood that we need to develop a phenotyping method that was reliable, accurate, but also fast, cheap, and open source. In fact, many of the phenotypic methods that have been used so far to measure seed size, they are based on manual measurements, which of course are labor and time consuming. And the digital method that have been implemented, they are not open source, or they are not parallelizable, or they are not flexible to implement other measurements, let's say color or other morphological measurements. So we wanted to find a solution to all these problems, and we decided to develop a new phenotyping method. So as I say, as genetic material, we use the rice global magic population, which was developed at the International Rice Research Institute by intercrossing 16 parents, which comprise 8 elite variety in the Indica subspecies and 8 varieties in the Japonica subspecies. And so at the time of the seed collection for our population, the self-crossing had reached the 6th generation. And of the 1,200 lines that were developed at that time, only 245 developed seeds in both environments. In fact, these reels were grown under 2 environments. The first one is under seedling stage drought condition, and the second one is under flooded well water condition. In particular, the drought was applied by interrupting the irrigation 35 days after sowing, and then afterwards it has resumed at different intervals. So as I mentioned before, we wanted to develop a method that was simple and cheap and fast to phenotype this collection for seed size and shape. So to do so, we first implemented a desktop scanner and a very simple setup, which included 2 color standards, which are essential to correct for light variation across the different scan, and a black tray, which is important to confine the seeds in a specific space on the scan. And so, considering both drought and well water samples, totally we scan 535 seed samples for an average of 100 seeds per sample. We account about 5 minutes to scan one sample. So considering that we have flexible working hour, we were able to obtain a scan in about a month. And then to measure the seed size, we decided to implement the latest a Python software called PlanCV, which has been recently introduced and comprise a series of modular and flexible functions that can be used to do different type of measurements, including shape. And so why we decided to use PlanCV? Well, first of all, it's open source and it's open development, but also because you can define your pipeline as you prefer, and you can include different functions in one unique pipeline. In our case, we just did morphological analysis, but if you're interested, you can also include other functions, let's say color, and you can keep updating this pipeline based on the samples that you have. So after defining the pipeline, we process all the images, and I use a common laptop, and the pipeline was able to generate all data in about 6 hours. So if we consider the time that we spent for scanning the seeds and then the time that we spent for analyzing the images, we can say that we got the data in less than 2 months, which is faster than previous methods. So here I'm representing very schematically what the pipeline does. And it's a simple image segmentation. So after uploading the image, the pipeline tried to correct for light variation through white balancing. And then I apply some thresholding to convert the image in a LAB color space. So in this way, we obtain a mask that can be overlapped to the image and then can identify object of interest in a specific region of interest. And then this object, that in our case are seeds, were measured one by one, obtaining 12 traits per seed. all these measurements are saved in a JSON text file that then can be converted in a final unique table CSV file that can be processed in any software. So here are the list of the, here you have the list of the 12 traits that we obtained. The definition are the one provided by the developers, and here at the bottom you have the link of the software. And so as you can see, they include the classical measurements of size, like say area, width, and length, but also like they have advanced measurements of shape and size. Let's say the solidity, which is shape measurements, but also ellipse eccentricity. These traits kind of express the roundness of the object. So it has value from 0 to 1, and value close to 1 indicate objects that are more round. So, but before proceeding to understand with our analysis, we wanted to be sure that our measurements were reliable. So we decided to resample and rephenotype a subset of 107 samples across both drought and well water samples. And here I'm representing the collinearity between the measurements for the same samples between the first and the second phenotyping. So as you can see, we had perfect collinearity for almost all traits. And so these indicate that our method is accurate and reliable. However, we saw that some of them had very low correlation index across the two phenotyping. In particular, ellipse angle had no correlation at all. And the width, even if the correlation is still high, 0.74, I think still indicate that there is some issue and there is not so much correlation, considering that they are the same samples that have been measured. So we decided to dig in this problem, and we went back to the definition of these traits by the developers. And here I'm trying to, there is a schematic representation of what the pipeline does to measure these traits. So based on the developer definition, length and width are the number of pixels that can be counted along the Y and the X axis respectively. And as you can see, based on the orientation of a rice seed on the scan, these two measures can vary across scan unless you constrain the seeds to have the same orientation, which will be very labor intensive. And the same is valid for the ellipse angle, which is defined as the rotation angle of this ellipse that is drawn around the seed. So, but at the same time, the pipeline provide other two measurements, which are ellipse major axis and ellipse minor axis, which are instead the number of pixels that can be counted along the measure and the minor axis of the elliptic shape that is drawn around the seeds. And we understood that these are better estimate of length and width respectively. So based on this, we decided to not consider these traits for the following analysis, but we also understood that this control was very important, and that even if there are many pipeline and tools available, it is always recommended to adapt and interpret their definition based on the biological species that you are dealing with. So for example, these measurements probably they will be accurate for other plant species, let's say species that have rounded seeds. But in the case of rice, if we will consider them, we will probably have conclusion that will be not so accurate. And so here now I'm going to describe more the variability that we observe in our collection. So I want also to say that on top of seed size, we also measure grain weight of 50 seeds per sample. And then we converted this weight to the one of 1,000 seed weight for convenience of comparison with the literature. So here I'm representing the total phenotypic variability that we can observe in the whole data set. And as you can see, more than 60% of the variability resides between drought and well water samples. In particular, these two groups are very different for seed area, major and minor axes, and for the perimeter. But at the same time, we observe expected correlation between the traits, especially those from classical size traits, let's say, and new size measurements, let's say area and convex area. They are perfectly identical almost, but also expected correlation, let's say the one between weight and all the seed size. These are expected and have been widely presented in the literature. Interestingly, we also see that some shape measurements, let's say eccentricity, is negatively correlated with minor axis, which I recall it is interpreted as seed width. So this means that based on the seed breadth, the seeds will have a less or a more rounded shape. But overall, we can say that under drought condition, samples produce smaller and shorter seeds as expected. However, we were able to see that some samples were outperforming under drought, or they were maintaining the same profile even if with less water. So here, for example, I'm presenting the trait values obtained under drought and at well water condition for the same samples. And the diagonal that you see, it's a one-to-one diagonal that was drawn in order to define a place where we can identify these samples. So the samples that are outperforming or that they are good will be above or on the line. So they will be in this left upper triangle. And as you can see, we saw that some of them were quite good. For example, here in the case of seed area, these samples are producing seeds that are bigger under drought than under well water, as for the case of the perimeter or the major axis and the minor axis. So we see that, so then we conclude that this method is reliable and also provided information about for the breeders, because these samples are candidate for drought tolerance in the following breeding cycles in rice. In particular, we observe this red dot. The red dots here are all the magic parents that kept popping up in most of the traits. And so we decided to see the variation for these traits among the 16 parents. Here you have the values for seed area in under drought and well water condition, and the samples that was popping out is this variety Shanxuan that is very popular in South China. As you can see, has a very consistent behavior across the two environment, and is likely that he's a drought tolerant, can be a drought tolerance donor for the future. And so then we decided, these are very recently results, but we aim to understand which is the genetic control of this seed variation under drought. So the same population has been genotyped in the past by my supervisor using genotyping by sequencing. In particular, we use two variants data set. The first one include variants that have been discovered on the Japonica reference genome Nippon Bare, while the second one includes variants that have been identified on the Indica genome 9311. And so here there are some results about the GWAS analysis that we perform for all traits in the two environments. And the dashed line here indicates the false discovery significance level of 0.05. And so as you can see, we find different peaks in the two environments, even if some of them are different. And to better see this scenario, this here is another representation of the Manhattan plot, but like seen from above. So here you have the chromosome where we identify the significant association, and these dots here are like the peak that you were seeing in the Manhattan plot, but from above. And as you can see, we see that some of these peaks were popping up out in both environments, like well water and drought, on chromosome 5, 7, and 12. But some of them were showing up only in one environment. And so on the other side, we also see that some of the common loci identified for seed size and weight were tightly linked to these loci, as is the case of GW5, GL7, and GW8. And also we observed that one single SNP on chromosome 5 was significantly associated with different traits, which are convex hull vertices, eccentricity, and minor axis. These traits are correlated to each other. In particular, as I said before, minor axis is negatively correlated to eccentricity, and at the same time is positively correlated with the convex hull vertices, which is another way of measuring shape. And so the identification of this multi-trait market trait association is because of the strong correlation between these traits. So we need to investigate more in detail these results and see which kind of gene are close or below this association. But we start to have an idea about the genetic control of seed size under drought. It is for sure a complex trait, but also what we see is that under the two different environments, different genes may be involved in the control of these traits. So here is the same results that we obtained, but with the Indica reference genome 9311. The results are basically identical, except for some association on chromosome 9. So in conclusion, what we can say is that our method is that is open source and flexible, is accurate and reliable, and allow us to better investigate seed variation in our global MAGIC population. But also, we understood that when applying we also understood that some of the traits that the pipeline provides are not recommended for seed size in rice. And so this suggests that when applying a phenomic pipeline, we need to understand the traits provided according to our biological samples. At the same time, we observed that under drought and well water condition, different genetic architecture may control seed-related traits. And so this is going to provide better information to the breeders for the future. In particular, we are going to see if these associations are due to a genotype by environment interaction. And so finally, I want to thank my supervisor, Brooke Moyers, which introduced me into rice and also the phenomic environment. And then I want to thank the students, Dani, which helped me with the phenotyping. I want to thank also the people at IRI that they performed the drought stress, and also the developer of Plant CV, which were very supportive and helped me with the pipeline. And of course, thank you for listening.
So thank you, Dr. Annarita, for your great lecture. It contributed a lot to our event and everyone who is watching. Now we will have 10 minutes for questions. If you want to do a question, please send it at the YouTube comments. I'm going to pick up the first question from Leonardo Volpato. Can you see that?
Yes. Hi, Leonardo. Oh, okay, so I have to read it. Okay, so he say, hi, Anna Maria, it's Annarita. I'm glad to see you and other friends here. did you take picture from both sides of the seeds to perform the analysis? Do you think that it could improve the results? Congrats. Well, this is a good question. No, we didn't take the picture from both sides. Also because I think dry seeds are kind of flat somehow. Compared to other species, like let's say if I'm considering maize, it can maybe useful to have both sides rice measurements, but no, we didn't, and I don't think I don't know, maybe we'll, but we will consider.
So I'm going to pick another one here.
Hi, Fernanda. Good afternoon. Is it necessary or done to measure the humidity of the seeds before the analysis? Does it affect image? Oh, well, the seeds that we obtained were already dry, and I think, yes, it's important. So we measure the seeds that they were dry.
Okay, we have another one here from Patricia.
Hello, Annarita. Thanks for the presentation. There are several types of images. When it is more suitable to use each of one for plant breeding. I'm interested in analysis of seeds images. I think she's asking about the type of images. It's not clear if she's referring to the extension or. So we took the scan, but I think so. We use a scanner which gives the normal RGB image, which are also the one that a phone can produce. But I mean, you can use different tools for that to do that. And about the extension, well, the pipeline doesn't care. You have just to define. So it is, I think what it was really important in developing the pipeline was to define the idea of the image, because of course, so we define a specific ID where we included the date where we were doing the phenotype, the type of scanner where we use, and also we were putting, let's say, 001 for the first scan that we were performing, and so on for that day. And then we, in the pipeline, we say, okay, the image, they have this structure, and these are the information that you are going to find there. And I think then, I don't know if there is, I think PlantCV can deal with many type of image extension. But the way you take them, it's up to you. You can also use a phone or a digital camera. I think probably the quality or like the resolution is important. Our images were 300 dpi.
She made another one that it's.
Oh, is it possible to measure insect damage on the seeds with images? Well, we didn't do that. I think you can do, so I think it depends on the kind of damage that the insect does to the seeds. For sure, the PlantCV, besides the morphological measurements, also allowed some color measurements. So I think you can also apply that. It depends on the kind of image. So, for example, if you are saying that the seeds is smaller because it's eaten by the insect, then you can use morphological. But if there is also a color variation in the seeds that they are eaten by the insects, then you can also use some color thresholding. And PlantCV has plenty of this function for do that.
Okay, thank you. Fernando.
Okay, there's a Fernando. It was possible to measure the seed vigor or viability by the image. Well, I don't know because they are dry. So we didn't consider that. I don't know exactly, but so I don't know exactly, but we will look into it. I think they are dry, so I don't know, but I don't know how we can do that, but it will be interesting to explore in the future.
Let's skip to the next one.
Hi, Annarita, congrats. Can this phenotyping be influenced by the presence of ons when estimating the seed length? Okay, yeah, this is a really good question. So actually, when I was developing the pipeline, we were using rice that we just grow in our greenhouse, and they were the ons. So we actually tried to see if we were able to remove the ions from the seeds, and you can do that by applying, as I say, the threshold with image color. So actually our pipeline can remove the ions from the seeds. However, the seeds that we had, likely they didn't have the ions, so it was not necessary, but yes, you can do that if you want to have a better length measurements. Oh, hi, Juliana. Annarita, do you think smaller seeds adapted better to drought as a strategy to germinate faster? Oh, I don't know, because I think during the drought, so the seeds I think is a sink organ where the plant accumulates starch to allow the seeds to germinate afterwards. So I think bigger seeds have more starch and they are more likely to germinate in the future because they have better resource. So I think, as a definition of drought tolerance, I would rather say that um, uh, like a tolerant plant it's a plant that is able to maintain big seeds to allow the reproduction and so to allow the progeny to develop. So I will say that yes, for sure, the smaller seeds is a way that the plant has to reserve water, but at the same time, I don't know if it guarantees a better germination afterwards.
So this was the last one. Thank you again, Dr.
Thank you.
for participating and answer the questions. So
Thank
we
you.
see you soon during the panel discussion
Okay.
later.
See you later.
Now, the second lecture will be given by Sara Tirado. She has bachelor's degree in agronomy for Iowa State University, and currently she is PhD candidate within the Applied Plant Science graduate program at the University of Minnesota. Her current research is focused on developing and implementing phenomics approach in maize fields to characterize the effects of genetic and environmental variability on plant growth. So, welcome, Sara.
Hi.
Thank you for accepting our invitation. You can start sharing your screen. Please select it here. Okay, go ahead.
Good. Thank you for listening to me. So I'm going to be talking to you today about some of the PhD work that I've done over the last couple of years. And really, over the last few years, most of my research has really been focused on really how we can use phenomic approaches to monitor both genotype and also growing conditions in maize fields. And I've primarily used very different sets of technology to do this. On one side, I have a lot of projects based on using RGB UAV imaging to look at how maize responds to different environmental conditions and also different management practices. And then on the other hand, I have a few research projects looking at how we can use hyperspectral imaging, and I'll talk a little bit more about that at the end and what it is, but how we can use it to monitor different genotypes and also to start to look at how we can detect and quantify different abiotic stress conditions. So most of my talk today will focus on more of the RGB UAV imaging part of my research because of time, but I will touch on a few things we've learned about hyperspectral imaging and basically some applications for it and why we would want to use it over traditional RGB imaging. So again, I'll start to talk to you about some of my drone work. And the motivation behind me really developing a project using drones was to really, although there's been a lot of progress over the last couple of decades, basically linking genomic information to phenotypic information and thereby making progress in selecting for optimal traits and making progress in crop improvement, we still don't really have a great understanding of how genotypic and phenotypic elements interact together with the environment. Also, although there's been a lot of advancements in the realm of genetics, we haven't really mirrored those advancements in the phenotypic world. So To do this and to start again to look into how we can start to track how both genetic and phenotypic elements interact with the environment, we can't really look at phenotypes at a single time point. And traditional methods of phenotyping, which involve a lot of work, a lot of labor, and a lot of cost, and a lot of time as well, basically are focused on measuring a single time point because of, again, all of these aspects. However, a better way to do this and to start to look at how plants are responding to the environment is to track phenotypic changes through time and throughout development. And in order to do this, we really need more fast and less costly methods of looking at phenotypes through time. And really, UAVs are a great way to do this because current commercial UAVs are pretty economical, so they don't involve large costs, and you can fly a field within just a couple of minutes and extract your traits. So some of my UAV work, the aims of my UAV work include developing a procedure for extracting traits using drone imagery. So that's kind of where I started, is how we can go from UAV imagery to certain traits that are useful, and then to use this pipeline to better understand how different genotypes react to different environmental and management conditions in terms of both in-season growth and also in-season productivity or yield. And lastly, kind of an unintended project aim from my research ended up being how we can use UAVs to track maize lodging and what the factors that contribute to lodging are. So again, I first started, I was given a drone and told to fly it over a field. And I'm sure most of you listening today have either flown a drone or know someone that has flown one and know that it's actually pretty easy to do it and it's fun. However, what's not so easy is going from that drone to that trait data that we want to use in biological applications. So that was kind of where I first started, is how do I go from drone imagery to some of this trait data? And so one of my first projects was to develop a pipeline to actually do this. And I'll talk to you a little bit about what the pipeline involves. But in general, we first fly our field. And actually there's a lot of open source software available for free to basically develop a flight map and path. And then it allows you to fly the drone automatically and collect images with a certain overlap. And the overlap is important because it allows you to then use structure for motion to actually be able to quantify the height of individual areas of your field. And so what each image ends up looking like is something like this. And how I like to think about them are like little pieces of a puzzle, and the whole puzzle is my field. So we then use software. We use Agisoft Photoscan software. It is not free. It does involve some costs. But we use that to basically grab all those pieces of the puzzle or my images of the field and put together the puzzle and create an orthomosaic is what we call it. And then from the orthomosaic, we can also, and again, using the overlap between images, we can find the height for individual parts of the field, and we can create 3D models. And from those 3D models, we can then create digital elevation models, which basically it's an image where each pixel just has one value, and that value corresponds to the height. From the orthomosaics, we can then extract individual plots. So to do this, we overlay a grid based on planting patterns, and we can then use the grid to subset a single plot that we're interested in looking at, depending on whether it's 1, 2, or 4 row plots. And then from that, we can then find our plant pixels and extract a number of traits. So some of the traits that we can currently get from the pipeline that I've developed are early in development. We can look at stand counts, or how many plants emerged from the soil. We can also somewhat track plant rotation, or the ability for corn plants to emerge from the soil and align themselves parallel to each other. We can also then track not necessarily flowering, but tassel exposure, which is slightly different than flowering. And then we can also start to take a look at some traits that change through time. And one of the traits that I've looked at is canopy closure, or the ability, and we do this by just finding plant pixels and looking at the ratio of plant pixels to total soil area, and then tracking how this changes through time. So the higher ability for the canopy to close in prevents weeds from growing underneath, and it also indicates kind of vigor and enhanced growth. And then lastly, we can also look at plant height and plant color. For plant height, it's been the major focus of my research and what I will talk about the most. And I won't get into the details of how we extract plant height, but if you're interested in knowing more, feel free to ask me a question. And also you can download this paper from PlantDirect, and it shows you the pipeline. It goes into accuracy measurements, and then you can also from there get access to my GitHub page and download the code to do this. So over the past 2 years, in 2018 and 2019 field seasons, I wanted to develop an experiment to start to collect UAV data and start to look at, again, the accuracy of some of the traits we're extracting, primarily height, and then also to start to answer some questions about how different genotypes are responding to environmental conditions. So we planted 12 different hybrids in replicates in 4-row plots. 4-row plots because we want to reduce the border effect and only measure plants in the middle 2 rows across the season. And then we did this across 2 different planting dates, a regular planting date and then a late planting date. It was typically around 10 days to 2 weeks later. And then we also did this, we also planted each one across 3 different densities, a low, a medium, and a high density. So we have a couple of treatments, again, planting date and density, and then we have 2 years for each data set. So every week in 2018, we went out and collected both drone data and hand measurements. In 2019, we collected drone data a lot more frequently because we found out that collecting data weekly missed some of the early season growth, which is very rapid for corn. But we still went out and collected hand data every week for 2019. So we also, at the end of the season, then went out and collected yield measurements to look at in-season productivity. So what this allows us to do is start to track, again, how different genotypes are responding to environmental conditions, in this case, years, and then also different management practices, like I mentioned, the planting dates and also planting densities. And so actually both years, this is showing 2018, but both years we had really strong windstorms that went through St. Paul and had very high wind speeds and actually impacted my field pretty drastically. So if we look at, for example, this genotype grown in the high density treatment, we can start to look at how we can track plant responsiveness to these environmental conditions, in this case, the storm. And we can see that, for example, if we plot this genotype across the 2 planting dates, there's a sudden decrease in height in the first planting date. And what this indicates is that actually these plots, because they're replicate plots, experience very high degrees of lodging because of the storm. And they ended up having a much shorter end season height compared to the second planting date. Now, when we look at our weekly hand measurements, we actually missed this. We can see that the first planting date had a shorter in-season height, but we don't know why. And this is because obviously if a field is completely lodged, we can't really go out there and measure height accurately. We can barely find plots until they're able to get back up. And also taking hand measurements requires a whole day of effort among many people, and it's hard to coordinate that from one day to the next. However, we can still just go out and fly a drone for 15 minutes. So using the UAV pipeline, we were also able to document differences in both growth rates among both growing conditions and also genotypes. So for growing conditions, this is kind of a very clear example, and there's more in this paper that's listed down here if you are interested. But for example, this genotype in the first planting date grown across 3 densities, we can very clearly see that plots grown at the highest density suffered from extensive lodging, and plots grown in the mid-density suffered from slightly less degree of lodging, but still significant amounts, and then the low density did not. So again, we're able to track how rates of growth are differing, and in this case, lodging are differing across our growing conditions. And then lastly, we can also look at growth rates among genotypes. So in this case, again, this is just another example in the 2018 season, but these 2 genotypes grown in the exact same growing conditions, so the second planting date and then the high density treatments, we can see that the genotype shown in yellow is able to have much higher rates of growth as early as 20 days after sowing, which is really early, and it's able to maintain this advantage and achieve a much higher final end season height compared to the other one. So this gave us the idea that maybe we can actually look at growth rates early in development and use them to predict end season traits such as height. So to test this, we took all of our plots, the height through time for all of our plots. We fit a B-spline curve, and it's basically a polynomial function of around 20 degrees. And we did this to create kind of a smooth curve representative of true growth. And we then took the first derivative, or the slope of that curve, which is shown here, the line in panel B, an example. This is just showing an example for a single plot, but we did this for all. And then we broke down that curve into 100 equidistant time points. We tested the contribution of every time point to end season height, and we selected time points that had the highest contribution. So 9 time points were selected, and these fell across 3 different developmental stages, ranging from V2 to V4 all the way to V8 to V10. And these growth stages actually accounted for that rapid early season growth that we see in corn, and also responses to that lodging event. So when we take those 9 time points and create a model to predict end season growth, end season height, we actually get very high accuracies. And if we break this down by treatments, planting dates, and then densities, we can see that we have, again, pretty high accuracy of an estimate for end season height by just taking these early developmental time points, except for the first planting date high density treatment, which actually is the treatment that suffered from extremely high degrees of lodging, and this likely affected our prediction accuracies. So as I've already kind of mentioned, we actually I went out to the field both years and saw a field that looked something like this, where most of my plots are laying down, and I was at first very upset. Actually, corn is really amazing in terms that even just 3 days later, the field will look like this. They end up goosenecking and getting back up, and it's pretty amazing. And this actually happened both years, so it gave me the unique opportunity to start to look into what are some factors that are causing this, and how maybe its ability to get back up can contribute to end season productivity. So something that I kind of want to point out while we're looking at this image is, you can imagine, so the field after this lodging event will always look like this. So there's really no alleys left. So you can imagine it's really hard for me to walk through this field. And a lot of research nowadays is actually geared towards developing ground platforms, such as robots that will navigate autonomously through the field and actually so basically go through and collect data, phenotypic data, or maybe administer some treatments. But you can imagine that if you had any type of lodging, you will no longer have the ability to navigate that field. However, you can always just fly a drone. A drone will never be impacted by elements such as this. So again, I wanted to start to take a look at the data set that I have in terms of lodging. So before I actually figured out how we can use UAVs to track lodging, I decided to first figure out, so how big of a problem is lodging across the US? And so to do this, I took the Genomes to Fields data set. So this data set is part of a kind of large collaborative initiative where universities and different agencies across the US grow thousands of plots, many genotypes in common. And then these are phenotyped for many different traits, including root and stalk lodging. And this data is made publicly available. It's done across many years. So it's kind of trying to create a large-scale data set similar to that which we can acquire through a company, for example, to start to answer some questions about G by E, etc. So this data set has root lodging mesh scored, as it's typically scored by just walking the field at the end of the season and manually logging how many plants have goosenecked. So that's how it's typically done. And so if we plot the percentage of plots within each year and each location that had more than 5% of plants lodged, we can see that in general, lodging is a huge issue. It happens every year. And however, it's very variable in terms of from year to year and also between locations. And that's kind of part of climate change, right? So we are getting increased incidence of high severity storms. And so this is only expected to aggravate lodging in the future. And then if we look at, if we break this down into plots that suffered no lodging, so less than 5% of plants actually lodged, and then plots that suffered from partial lodging or extreme lodging, partial lodging referring to plants that have between 5 and 50% of its plants lodged, and then extreme lodging more than 50%. If we look at the yield data for these plots, we can see that you can get up to a 12 bushel per acre decrease when we compare plots that did not lodge to those that did suffer from extreme lodging. So this is a huge deal, and I'll note that, I mean, so these yields aren't what's typically observed in a production field, and still 12 bushels per acre is a huge decrease. So you can imagine that in a production field, this would probably be even more. So back to my data set. So what I wanted to kind of start to get a handle on is how we can use UAVs to track lodging. And so I had two data sets, and the time point in red, and the drawn in red up here corresponds to the time point where the lodging event occurred in each year. So you can see that it occurred at different time points, and in general, but it happened both years. And I was able to get out and fly consistently right after the lodging event across both years. And what we can see when we look at our 3D models, you can clearly see areas of the field that have experienced lodging. This is the border road, for example. And then the other ones correspond to very specific plots. If we look at the difference in height in those digital elevation models that I mentioned, the day before the storm, so we'll be taking the time point right before the lodging event, to a time point right after, we can see that there's also clearly areas in the field that experience lodging. So the metric that I decided to come up with to study lodging responses was the percent change in the average UAV-derived height the day after the storm to the day prior to the storm. So it's one number that we can extract for every plot, and then that way we can compare across plots between the different years. And so what this looks like for my entire data set, so this is showing data from all three densities, both planting dates, and both years, so 2018 and 2019. And this is a density plot, so this is showing, for example, the plots that suffered from, so everything below 0 suffered from lodging because the height decreased after the storm. Everything above it did not suffer from lodging and actually got an increase in height. And so, for example, if we take this plot over here, this orange dot, this plot did not suffer from lodging. It kind of stayed consistent. It didn't increase, but it didn't decrease either. And then this plot over here actually suffered from extreme lodging, the one shown in blue. So if we break this down into the different environmental conditions, so if we break this down by both years, the 3 planting densities, and then the 2 planting dates, we can see a lot of variability in our lodging responses. And what's actually pretty cool is that, for example, in 2018, the first planting date suffered from most of the lodging. However, in 2019, the opposite was true. Now, when we look at the developmental time point that these plots were at, they're actually very close in development. So the plots that lodged in the first planting date were at around the same growing degree units as the plots that lodged in 2019. And actually plots that were either slightly younger or slightly older were not as affected. So this kind of gave me the idea that maybe there's a few factors that are impacting our lodging responses, including both what height the plant was at before the lodging event occurred, and also the growth rates of those plants early in the season. And I got the idea of the growth rates because we can also see that there is an increased incidence of lodging at higher densities. So these plots are likely growing faster and then likely experiencing higher degrees of lodging. So what I did was, again, take up my height curves for each plot and then extract 2 metrics from this. So I took a time range early in development, and then across mid-development, but prior to the lodging event. And I extracted the slope of these 2 time points. So I ended up with a metric, which would be rates of growth for early season and mid-season time points. And we used this together with many different variables to see which variables are most predictive of lodging responses. And what we found is that both the planting density, well, not both, 3 things, the planting density, these 2 metrics, early season growth rate, and then also mid-season growth rate, during these early vegetative stages, were all the most predictive of the lodging severity that these plots experienced. And if we create a model using these variables, we can get very high prediction accuracies for our treatments. So there's a lower prediction accuracy here because, as you can tell, these plots did not lodge. So we also wanted to take a look at what the yield impacts would be, and we kind of found the same thing. So plots that were that experienced higher degrees of lodging had more variable and in general much lower yield. So there's a significant difference in yield across the 3 treatments. And again, you can see that severe degrees of lodging also experience a lot more variable yields. And if we break this down by densities, we can kind of see the same trend. So plots at higher densities also experience more plots that had severe lodging, and the yield was also more variable and lower compared to plots at lower densities. And again, if we look at what variables are most predictive of yield when we're taking into account lodging, we can see that the early season growth rate, so it would be that first growth rate that we extracted, not the second, that was the mid-season, and then also the lodging severity, we're able to very accurately predict the yield that we were getting across our different plots. So some takeaway points from my UAV work that I want you to take away are that first, we can use we need temporal phenotype estimates to better start to understand how genetic and phenotypic elements interact with the environment. We also can use plant height UAV measurements to start to document differences in growth rates throughout the growing season across both genotypes and also different management conditions. We can also use early UAV measurements to start to track end season traits. So in this case, we used early season time points for height to track to predict end season height with pretty high accuracy. But again, something similar could be done for other end season traits. And lastly, that we can use UAV plant height estimates to start to look at variation for lodging responses. So that's kind of my part for the UAV work that I'm going to share with you today. So now I'm going to talk to you a little bit about some of my work with hyperspectral imaging. So this is so when I first started my PhD, I was in charge of growing a lot of different plants, and we had many different genotypes growing in our growth chambers as seedlings. And then we started exposing them to different treatments. And so really from very early on, when I look at 2 plants, it's really hard for me to actually tell what genotype I'm looking at. Most genotypes look pretty much exactly the same. Also, when we look at the different treatments, you can tell that they're kind of stressed in some cases. In other cases, it's hard to tell whether they're stressed or not. But it's really hard to tell apart which stress your plant is experiencing because the symptoms are actually really similar. And also, it's hard to start to quantify. So is this genotype more stressed than the other? I don't really know. So again, if I just gave you all of these RGB images, it'd be really hard, at least it is for me, and I worked with these plants for a really long time, to tell which genotype is which and also what stress we're looking at and how stressed the plant is. So how I like to think about RGB imaging, which is similar to what we can see with our eyes, is that it's kind of colorblind. So there might be actually things going on within the leaves, such as biochemical changes, that we're actually not able to capture by just visual imaging. So how I like to think about hyperspectral imaging then is as it being able to capture some of those things that we can't visually see. So hyperspectral imaging is able to measure many different wavelengths with very high affinity. And so we can maybe then start to see, again, things that like biochemical changes that are going on inside the plants that we're missing with some of the RGB imaging. However, when we image a plant with a hyperspectral sensor, so this is actually an example from one of my plants, we use kind of a top-down approach. So you're looking at the plant from above. For each pixel, we actually get around 300 values for 300 different wavelengths. So you can imagine that this is just a small part of an entire image, but you have this gigantic data cube. And data can accumulate very fast. There's a lot of data to deal with, and it can become extremely challenging to do this. So what a lot of people have actually done is to first just find the plant pixels, which is pretty easy to do. You have a lot of wavelengths. You can figure out which pixels using NDVI or some other metric are of the plant. And then they grab all of the pixels from the plant and they extract an average for each wavelength for the entire plant. So what this looks like, an average would be, for example, this line right here would be the average for all the pixels in this plant across the different wavelengths. And when we look at the deviation in the variation in reflectance for each wavelength, we can see that there's a lot of variation that this average is actually missing out on. If we look at a single wavelength, in this case, just looking at 625 nanometers across the plant, we can start to see that there are areas that have much higher intensity in reflectance compared to other areas. And for each wavelength, the pattern that we see across the plant can vary. And so this kind of brought us to think that, okay, so if we are extracting an average, yeah, it's a lot less data, it's a lot simpler to deal with, but we're getting rid of all this variation that can be kind of hinting at some of the changes in biochemical properties that also vary spatially throughout the plant. So this brought us to design an experiment to start to think about a few questions, including how much spatial variation in reflectance we can observe within plant leaves, and then also what the ability of reflectance data for different regions of the leaf. So if we take the center of the plant compared to the tip of the leaf, for example, what the ability for those regions are to resolve between different genetic and environmental factors relative to extracting data for an entire plant. So to do this, we came up with an approach, and again, you can look up this. There's a preprint online in bioRxiv, and I have the code in my GitHub page to do this, so we can feel free to download it. But we can find the center of the plant, and then find the longest leaf, break down the leaf into equidistant segments, and then extract all the reflectance values for each segment along the leaf. And that way we can compare reflectance for different regions of the leaf, such as the center, the middle part of the leaf, or the tip. So the hyperspectral sensor that we use, again, it's a top-down approach. So the sensor is up here in a platform. It looks like this, surrounded by lights. It's all indoor-based for now, but the idea is to then start to apply it outside. And then it's a line scanning system, so it images the plants this way. So we imaged many different genotypes grown across 4 different treatments: the control, salt stress, cold stress, and also heat stress. And we imaged the plants using the sensor right before the stress was applied, right after they came out of the stress treatment, and then a few days later. So again, the goal of this was to start to look at variation in reflectance throughout the leaf, how it varies, and how different regions of the leaf could help us in maybe identifying what stress we're looking at, what genotype we're looking at, and also start to quantify some of the stresses. And what we found was when we look at variation in reflectance throughout the leaf, there's evident patterns, and these are consistent through time. And the patterns differ in terms of intensity, and these can be indicative of or useful in when we're trying to look at other things, such as assessing how a plant is responding to some environmental condition. And again, this also indicates that maybe then taking an average for an entire plant is not the best approach. Then using this, again, variation between the different leaf segments, we were actually able to distinguish between different maize genotypes. And what we found is that the ability to distinguish between these genotypes was actually due to morphological differences across the genotypes. So genotypes that are morphologically very similar, so like things like leaf angle, how green their leaves are, how fast they grow, etc., how tall they are. All these things impact the hyperspectral data that we're getting. However, it is able to maybe not tell apart genotypes that are extremely similar, but able to cluster them into genotypic clusters that are pretty similar morphologically. And clustering them into these clusters is actually necessary if you want to then start to take into account things like abiotic stress conditions. Otherwise, changes in morphology between genotypes kind of outweigh the more minor changes that we are observing in more of the biochemical properties within the leaves in response to some environmental events. And then after clustering them within genotypes, within each cluster, we're able to pretty accurately detect different abiotic stress conditions. And the cool thing we saw about this is that for different stresses, different parts of the leaf are more informative and more useful. So then that's something that people can take into account. So maybe I don't need data for an entire plant because that's a lot of data. I can just subset a specific part of the plant that will be useful for some other application. And lastly, we can also apply the same method to start to quantify how stressed the plant actually is. So that's really all I'm going to talk today about hyperspectral imaging. But feel free to ask me any other questions at the end. And I think first I'll acknowledge some of the people that have helped me through my PhD and this research, my advisors, Nathan Springer and Candy Hirsch, a former postdoc, Tara Enders, and then current postdoc, Shakai, and then Susan helped immensely with growing the plants, figuring out how to apply the stresses and all of that work. And then these are just some funding agencies that have provided funding for this work. And with that, I'll take questions.
Thank you, Sara, for your great presentation. Now we have again 10 minutes for questions. I have just select one here. I will put it on the screen for you.
So I'll read it. So, hey, Sara, nice to see you here too. Do you attribute the worst correlations between ground nodes and UAVs due to unreliable ground nodes? Yeah, that's a great question. So actually, yeah, 100%. So what we found out was that when we correlate, for example, our measurements to when we start to correlate our UAV measurements to our ground measurements, how we get our ground measurements is we measure by hand just 2 plants per plot because there's so many plots, and if we start to measure all the plants, we'll never finish. And yeah, so we measure 2 plants per row, and that's typically done in the industry as well. And then what we found is those correlations are pretty weak, but they're actually pretty much the same as the correlations when we take 2 different plants within the row and measure them to 2 other plants within that same row at the same day. So there's a lot of variation actually in height within a plant, and maybe within a row, sorry. And then also like how we measure it, there's some measurement error, inherent measurement error in that. So even measuring the same plants manually will give you very low correlations, kind of the same correlations as you would get from our UAV to our ground measurements.
So I will pick one more another. Yeah.
Hi, Sara. Do you think it is easy to use the same approach to other crops which present lodging problems, such as soybean? Thanks. Yeah, absolutely. So because we're using height when a plant lodges, either and also within corn, you can also use this to assess things like stalk lodging. The difference is in stalk lodging, the plants won't get back up, but you can assess that rapid change in height and then assess how many plants lodged and correlate, and also look at kind of correlate how many plants lodged to something like end season productivity of the entire plot. And of course, you can do the same with pretty much any crop where lodging mainly causes a decrease in height. Hi, Sara, great job. Is better image quality, higher overlap, or lower flight necessary to assess height and lodging when compared to traits related to color, such as maturity? That's a great question. So really, it depends on your plot size. So if you have, say, 1-row plots, you're gonna want better image quality in general in terms of resolution. So where your pixels represent less ground area compared to having, say, lower resolution would be pixels that represent a larger area in the ground. So you just want to make sure that you have substantial pixels within your plot that are represented in your orthomosaics. So that also relates to kind of your flight height. You don't really need super dense clouds to get a good elevation model for your plot. However, you do always need high overlap. So I always recommend at least 85 front lap and also side lap. So overlap is the biggest contributor to height, basically. For things like color and maturity, then you probably also want kind of better image quality, and you might want to fly lower so that you can also look at variation in color throughout a single plant rather than throughout an individual plot only. But yeah, overlap would be the main contributor for height.
We have one more.
Hi, Sarah. What is the best software to construct orthomosaics? So I am biased. I love Agisoft Photoscan. Like, I think now it's actually Agisoft Metashape. But the other one that's traditionally used is Pix4D. They're both equally good. I've seen a lot of studies that compare the quality of the orthomosaics in both, and I like both. In my opinion, Agisoft can give you a little bit more flexibility in terms of parameters when you're constructing your orthomosaics. So how you stitch your images together, you can kind of control it a little better with Agisoft, in my opinion. Maybe someone that works mostly with Pix4D will tell you the opposite, but they're equally good in general.
Okay, this is it. Thank you, Sarah. It was a great presentation.
Thank you.
See you soon during the panel discussion.
Okay, thanks for listening.
So now we will have a quick break. So see you in 10 minutes. So, hello everyone, we're back with our lectures. Now, the next speaker is Dr. Felipe Matias. He has a bachelor degree on agronomy from Federal University of Uberlândia, master degree in genetics and plant breeding at Paulista State University Júlio de Mesquita Filho, PhD in genetics and plant breeding from the University of São Paulo, and he is currently postdoctoral research science at the University of Wisconsin. He develops works in the areas of quantitative genetics, genomic selection, association mapping of polyploid species, remote sensing, and high throughput phenotyping. So, welcome, Philippe.
Hey, can you hear me?
Yes, I can hear you. I will share your screen now.
Okay, I'm gonna.
And you can go ahead.
Yes, well, guys, thank you for inviting me for this presentation. I'm really glad because 2 years ago I was a member in GEVANC and then I could learn a lot. And then now I'm here as a speaker. So thank you so much. Also, thank you to doing this symposium or seminar in English, which go over country. You can have more access for different people in different countries. I'm gonna try to do my best here. Okay, please let me know if I'm talking too fast or if sometimes I need to slow down. So my idea here today is talk about my research and my experience with remote sensing plant breeding. I start last year, so I have only 1 year of data, and then I prefer talk today more about applications and how we are thinking to use this data back here in Madison, our potato breeding program. Okay, please feel free to making questions. We're gonna talk. So I pretend to start talk about our potato breeding program, and then start to talk a little bit how we can use information that we collect with remote sensing during the sprout stage, and also vegetative stage and senescence time. I had more information here before, but for 30 minutes I need to remove a lot of information, but maybe we can discuss a little about flowering time, a little bit about flowering time during the discussion panel section. So let's start explaining how the UW potato breeding program at University of Wisconsin works. We have 2 agriculture research stations. One is right here in Rylander up north. Another is in the middle of the state, which is Hancock Agriculture Research Station. And as you can see, we have different soils. So we have these 2 environments in our state, okay? And we started the breeding program for the crosses. What we are doing right now around 100 crosses for all category, market categories that we have. So we make our crosses, and then we have the seedling stage when the plants go growing in greenhouse. Basically, these 2 stages here, it is in a greenhouse. And after we do some selection for seedling, we have our first year in the field. Okay, this first year in the field, we have only 1 plant, as your normal breeding program, guys. We have only 1 plant, and we should evaluate that that field year for traits with higher heritability. For example, for example, maturity, shape, size, and skin set. Normally, the breeder go walking the field and doing visual selection. We are not doing measurement. We are not collecting yield or weight, okay? And then we come for field year 2, that also is in the field. And then here we have 8 plants per plot. And we maybe, for example, for the chips population, we can evaluate color, fry color, or dry material. But in general, also field year 2, we are doing visual selection, okay? So just keep in mind that field year 1 and 2, we are doing visual selection here. And these 2 years fields, we plant over Rhinelander up in north, because we have better conditions for keep the production of seeds and disease problems. Then from year 3 and 4, we already have much more potato seeds, tuber seeds, and then we can have bigger, greater plot size. And then now we starting measure traits in the field, like putting the potatoes in boxes and weighting them. And with this information, we can have an idea about yield. And then from either year 3 and 4, we also start having some trials up south here in the center in Hancock Agricultural Research Station. Okay, so 2 environments. Field year 5 and 6, we start our national trials. Which is these national trials? As Sara said before, the breeding programs here, actually the potato breeding program here, they have connection with different states, and then we receive their genotypes that also are in field year 5 and 6, and we also send our genotypes for other states. So we kind of improve the number of environments that we are evaluating, okay? And then we continue, in around 10 years, we are releasing cultivars. So this is the main pipeline that we are using up here at UW Madison Potato Breeding Program, okay? Just keep in mind, field year 3 and field year 1 and 2, we are doing visual selection. We're gonna talk about that later. So we have different market categories, but today I'm gonna talk about 3 of them. We have the chips, the reds, and the russets. Because, guys, I need to share one thing with you. Reds, Brazilians, when we are saying R, it's kind of hard. And then sometimes I say heads, which means head. And nobody understands, but reds, okay? If my boss is watching this, he's gonna laugh about that, because I always say heads potatoes. Okay, anyway. So here I'm highlighting 3 main varieties that the UW Madison Potato Breeding release. We have here the Hodag, which is potato chips. We have the Red Prairie, which is the one cultivar from Reds. And also we have the Plover, which is the russet. Okay, so potato chips, this is a fresh market, directly fresh market. The russets you sell also for fresh market and also for fry. Okay, so what my job is here, what I'm doing right now, I go in the field and I collect pictures during the entire crop growth cycle. So I start collecting pictures actually before I start the sprout, and then during the sprouting time, vegetative time, flowering time, and maturity. And I'm gonna give today some examples how we use the information that we collect in each one of these stages directly in our plant breeding. Easy approaches, actually. Let's start here during the sprouting period or stage. So as you can see here, in this moment during the sprout, we can evaluate the number of plants per plot. This is the pictures that I have this year. We took one set of pictures on May 27th and also June 2nd. The difference here is 6 days, okay? And as you can see, in 6 days, how the plants can grow faster, right? And we want to count them. And for do that, the approach that we're using, it's called image segmentation. And as you see, Annarita talked about image segmentation when she was removing the background from the seeds. Sara also talked about image segmentation when she was selecting only the plant in that background. And then here's exactly the same. As you can see, we apply one mask. Both of the 2 guys also talk about the mask. And we remove the soil and we keep the plant. We are using for do this one package called Field Imager, which is a software in R. And the function that's related with this process is called Field Mask. Okay? So basically what we do, we calculate one vegetation index. In this case, we are using the Hui, the Hui. And the Hui said to us, the black spots, the low values, is related with plants, and the white spot, the white region, is related with the soil. How we do this better? One approach, it's called threshold method. The threshold method, we take that layer, the vegetation index layer, and we plot the values here. And then it's possible to observe in this specific case, two peaks, okay? There is this small peak, which represent the plants, and also this huge peak, which represent the soil. But remember that index that we use, the Hui, this one here, it is specifically to identify soil and greenness color. And that's why we don't have like another columns here. But it is possible to have, okay? As you can remember here, there is this all other values around. So what do? We come here, we set a point, a threshold crop point in 0, and we say everything above this value is false. We wanna remove the soil, and everything below 0 we wanna keep because it's true. It's our plants. This way we make the mask. Okay, does that make sense? Okay, and then with this mask, we can use another function for the same software called field count. And this function basically use one algorithm called watershed, which help us, it's pretty common in image segmentation actually, help us to identify each individual points here, each individual shadows here, okay? And why it is important during the take more than one set of pictures to use this approach? Because, for example, for early genotypes in May 27, they didn't come out. For example, this plot here, they had only 5 plants. And these other plots here have no plants at all. And 6 days later, we can see 13 plants in the first plot, so it increased from 5 to 13, and also 12 plants in this first plot that was 0. This method is important because when you have a breeding problem like our case, we have early and late genotypes. So if we go to the field and fly the drone only once, maybe we are losing a lot of information there. And then you have some results like comparing the visual score and the remote sense information, say, oh, actually, it's not work. So actually, you are taking your picture maybe in a wrong time. You should have more information. But look this. We also have some problems here. For example, this plot here, the first slide, we are talking about one genotype that has early maturity, grow fast, and we count 15 plants. And each plot here, just for you know, we have 15 plants. So this is the right number. We have 15 plants, but in 6 days later, the plants already start touching each other, as we can see here, right? The plants start touching each other, and then the algorithm from image segmentation stopped to work. They didn't work well, reducing for 11 plots. Here's another example, reducing from 11 to 9. So if you want to use this approach, you should take maybe more flights. It helps you to evaluate early and late genotypes using this approach. And also, you should select the values in these all columns that we are extracting. You should select the values close to the 15 or whatever number of plants that you put in your plot, right? This is only if you are using image segmentation, but there is over there many different software that use machine learning to identify plants. But the softwares, sometimes machine learning models, they need that you train the model with many examples. It can be kind of hard to do this for crops that's not so common, like corn, soybean, cotton, or potatoes. you can maybe, this is only one suggestion for you to start doing this, okay? Also, potato has this really nice shape, and then when we look, the picture is just one shadow. But when you come for the gramíneas like maize, we have this leaf over here and this other leaf over here. So when we look the shape, maybe the algorithm can count 2 plants here, 1 plant here, another plant here. So the people that is working with grasses maybe should find the better moment to start taking pictures. For example, when the leaves stay together. When you use vegetation indices to select what is green, it's pretty precisely to find this. So maybe you can start using this time here, or maybe you can develop some approach and divide the number per 2. Just giving some ideas. There is a technology over there, but you need to choose whatever is better for your breeding program. Okay, so this is the first application that I want to talk about, counting number of plants. Let's continue going here in our crop cycle, and then we are here in the vegetative growth. I want to start talking about vegetative growth, talk about how important is this in plants. I'm gonna describe better in potatoes, but you can try to figure out and associate this with your crop. We have the phases here during the crop growth cycle. There is the phase 0 that goes from planting to emergency. Okay, phase 1 goes from emergency to tuber initiation. Phase 2 go from tuber initiation to end of leaf growth, what means this? The plant already gets the higher leaf production, and almost 90 to 100% of all energy or all assimilates that the plant's capturing going directly for the tubers. Okay, and then after this, because we come phase 3, which is because all energy is going directly for the tubers, the plant starts dying and then complete die here, when is the end of the crop growth cycle, right? The dotted line means the solar radiation intercepted by the crop. As it's obvious, there is no observance of radiation before the emergence. The plants are under the ground. And then when the plant starts growing, this goes super fast. And then the plant has a genetic control there is a genetic control, and also each plant has a specific size where you get your maximum of leaves and size. And then after this, the plant starts only not growing anymore, but starting receiving. I'm talking about some specific kind of potatoes here. With annual crops or determinants growth. And this draw line means how much of the assimilates is going from the plant to the tubers. As you can see, also goes the energy every time is going more and more for the tubers. In some point, like here, almost all energy and assimilates go only for the tubers, and then the plants start dying. Okay, so why this is important? This process here can be different depending of which genotype we are saying. For example, the first genotype, G1, they get first for this environment, for this specific environment, they get first in this maximum production or assimilation, energy assimilation, and they stay theoretically more time sending information or sending this assimilate to the tubers. Okay, this other genotype here, genotype 2, they had they took more time to get here, and they have less time to send this energy for the tubers. And also genotype 3 is super late genotype, and they stay less time. And then you can think, okay, maybe genotype 3 is late, but if we leave them in the field, maybe it can produce more than genotype 1. That's true. But what's happened here up north? We need to kill the vine at some point. because we need to start our selection process. And also the growers, the farmers, they also need to kill the vine at some point because the winter's coming, right? And then when the winter's coming, they need to have everything already, harvest and control. So this genotype tree actually had less time to send this information or the simulates for the tubers. Okay, so I expect that you guys agree how important is this phase 1 for relate with yield. So how we can use this information in our plant breeding? Using remote sense information during this stage in our programs, okay? First, let's start to evaluate some vegetation indices. Here you can see there is a lot of vegetation indices. Any DVI and any DRE is pretty common indices that we see in the literature. But today I select the GLI, was randomly. Here's the equation that it's related with GLI, and I'm going to talk about this equation later, and also it's related with chlorophyll or greenness traits, okay? And to evaluate green index, we use the field imager function called field index. Another trait that we also can collect during this time is the estimate canopy. As you remember there, the canopy is related how fast or how this potential of the plant grow during the phase 1 that can be related with yield at the end. How we are evaluating that? Basically, canopy is related with the area where the plants are covering the soil. You can give this information in 2 ways. We can give the area of plants per plot, like this example here, which means that 62% in this plot is represent with, we have plants over there. Also is related with leaf area index, right? But we also can give this information by proportion, which means we took, for example, in this example, we have one genotype that covers 70%. We can say this genotype is number 1, and the others is relation to this main genotype. So you can give this information per plot or per plant. In this case here, I took the way that comes from this function, field area, in field imager, which is related per area. Okay? Okay. I say a lot of okays, I know. Let's continue here. So what do we have in this image? We have the x-axis, the canopy that we just discussed about, collected 40 days after planting, which is that specific time during the vegetation growth cycle. And we observe here on the y-axis the total yield that we have. So what we did? We just did a simple regression here and to observe how related is our canopy with our total yield. And as we can see, our commercial cultivars, they are concentrated in this region, right? And all these blue points here is the better genotypes for total yield, is our best genotypes. And we have a coefficient of 0.35. As you can see, we have some outliers, but here, again, I'm just showing how you can use this information. But when we observe the heritability, the canopy is explaining 91%, which is amazing, right? And when we look total yield is 79. That's great. And what's the idea? The hypothesis or the question is, can I select one point of canopy during the early stages that is related with yield, and then I can apply this in my program? That's the question. For example, I know that genotypes that normally has greater canopy, like you can see here, they also have a better yield. How we can apply this in our breeding program? You remember that I was talking about field year 1 and 2 that we normally only do visual selection. So we are not collecting this information in our field. we are only selecting based for higher-order traits, for example, skin set, color, shape. But imagine if we can also use this information from the kernel that also is related with yield to give us better confidence in our visual selection. That's one thing that we are going to start in doing this year here. So as you can see, how we do this process in our potato breeding program, we harvest, and then we have here one plot with a sample, okay, and then other plot with a sample. And when the breeders, here's Dr. Anderman and my colleagues, when he look around and say, select this genotype, we go there and put all seeds that we have in this entire plot in the box. So we have here one selected, another selected, and here not select genotypes, okay? And here how normally the field looks like after we finish. That's good because we can bring potatoes home, and so our mom always loved potatoes. So the idea that I'm suggesting here, you can take this information that you took in the early cycle, during the entire cycle, the same thing that we did for the first stage, we can do during the flowering time, and we can do during the maturity time. If it's this related with yield, you can put this information in your phone, your tablet, and when you go to the field, you look the major traits, again, maturity, shape, and color, or some disease, but also you can have an idea, the breeder can have an idea what the yield that genotype is supposed to have. That's one of the second application that I'm going to talk today. And I guess it's pretty easy to start using this in your breeding program, I believe. We are today, actually this year is going to be the first year that we are doing that. And then next year, let you know if it works. Hopefully it works. And then we come here during the senescence time. As you can see, one important trait for potatoes in this point is what we call vine maturity. Vine maturity, we go to the field and we score our populations from 1 to 9. 1 for early maturity, when the plants are already dead, and 9 late maturity. Okay? If we are observing here colors, we can use vegetation index that is related with color. For example, in this image here, we are using NDVI, right, which is the proportion of the reflectance of near minus red divided by their sum. How it works, from the 100% of general, okay, I'm gonna talk in a general way. From the 100% of the radiation that the plant receive, the plant reflect 8% of red in this case, and 100% energy come, and the plants reflect 50% in the near spectrum. As you remember, Sara was talking about the spectrum. We are talking about these two specific regions. And there is this huge difference between when the plant is green and when the plant is brown or dead here, because the plants reflect more at the red region. And we can see that any DVI for green plants normally is higher than any DVI for maturity or late or dead plants. Okay? We have one trait that we can associate with maturity. Another trait, as you can see, when the plant is green, is tall, and then according to the plant go dying, the plants reduce the size. Thinking on that difference of height, we can use, for example, estimate plant height. Sara also talk about the estimation, talk about how she used that for logging, and now we are talking about the same trait that we are using for maturity. How we are collecting these? To estimate plant height, we use two files from the orthomosaicing step, which is the digital surface model. In potatoes, we took the first flight before the plant starts sprouting, or after the healing, as you can see here, and then another flight when we have plants. As you can see, we have this green part is the plants, is the higher. And what do? With these two informations, we subtract the base from the plants, and then we have the canopy height model. Basically, if you see all this region, the green region here is higher, right? And then when we extract it, and then if you not extract the information, all these plants that are here, probably they're going to have higher height, and we are going to introduce a bias in our data. When we extract these base from our real pictures, we put everything around 0, and then we have only the plant height. Okay, does that make sense? I guess. And afterwards, you can extract the data, the average. How it looks like. Again, let's see here in this graphic. The x-axis, we have the vine maturity score. So we go to the field and we use our sensors, which is our eyes, to identify plots. And if you remember, our scale goes from 2 to 9, right? Here I'm using the reds. And here's the index value. As you can see, the NDVI had better score coefficient, and the correlation DVI maturity was 71, which is pretty amazing. And also we have the heritability, 63. And these also we have for the other index, GLI, that we already talked about before, and NEGRE. And also here we have the height that we are evaluating. And see, the height is 74% heritability. We are explaining at the last moment in our growth cycle, 74% of our heritability. And all these values are higher than the actual heritability for vine maturity, which was 0.5. We were kind of expecting that because the index, they are much more precisely to get individual pixel information and averaging that to for the entire plot. And here we are using our own eyes, and maybe my sensor is different of my colleague's sensor, and then what's green for him or for them is different from what I guess is green, and then I can introduce some bias in my data. So it is important, and it is super cool work with that. Okay, the idea is change from manual selection, manual evaluation for using tools to evaluate for us. How are we gonna make sure that these tools is really related with the same genomic region? Because, okay, the correlation is high, the heritability, we are explaining a high heritability, but how I know that actually we are selecting for the same genes? Let's try do genomic association. Using the same data that we talk about the reds, this population, I have 7 families, right? 93 genotypes and around 9,000 molecular markers, and we are using SNPs here. And as you can see, we have here the 7 families. This is the kinship matrix, the relationship matrix between individuals using the molecular data. We can see that all the siblings, we have a structure because, of course, we have siblings. They are more related genomically, and they're supposed to clusterize together, as you can see here, right? So we have molecular data. We have phenotypic data that we collect in the field with the drone. Let's try run some GWAS. I'm using here the GWAS Poly, which is also a software in R, and I'm using the U model, which is this one. We can include the fixed effects. We can include the marker effect for this position. Can be additive model, dominant model, general model. It's pretty amazing. You guys should go and look this paper. And also we should correct our variability in function of the structures that we have there. In my case, I'm using the kinship matrix here in this portion in my model. But okay, let's see some results. This is my Manhattan plot using estimate plant height. I choose plant height because we already talked about index before, and I want to show some different use. And we found this really strong peak in the Manhattan plot here in chromosome 5, as you can see. And we have this we have this marker here. I guess the potato guys that know which mark is this and know this region know what I'm talking about. I'm gonna, and maybe you guys laughing now because you can see that this trait is related. I'm gonna talk this. And here's the QQ plot, right? So all my markers is following what we are expecting, and then at some point we have this increase, which means that there is some information in that region that actually is related with the variability that I'm looking in my data. So let's look closer to chromosome 5 and what you have there. Here is our markers. The marker that we found significance, the marker that had higher significance is this one here. And right here is the CDF1 gene. CDF1 gene is a pretty common, many is already identified in many species, and also this gene translates for transcript factor that's related with the flowering time, tuber initiation, and maturity. The regulation of this gene is related with the maturity. What we are doing here, we have a physical proof that what we are observing in maturity is also observed with the data that I'm collecting with my drone. We are proving that, which is amazing. This is a really simple example, but when you think in traits that is expensive to evaluate, for example, biotic stress, in this picture, we were evaluating PVY, which is a virus, and we use this kit to evaluate if we have or not the disease, and it's kind of expensive. If we can exchange or change the way that we're evaluating for that for a tool that we can fly in the field, it's super amazing, and we can run time. Okay? All this information that I talk with you here today is in my GitHub page. I have this tutorial there for the software field imager. There is many examples. You guys can go there, download, follow the coding, and send your feedback. There is some videos over there. Let me know if you're liking or what I need to improve. If you put your cell phone in this QR code right here, it brings you guys directly to the web page. So let's continue talking and have this interaction.
Thank you, Felipe.
Yeah, I'm gonna just say thank you for you guys. And then
okay.
we can start our questions. I told you.
Well, let me finish.
Yes, awesome. So I wanna first thank you for Gevenk, because 2 years ago I was a student there and we organized the symposium. Here's my actual boss, Jeffrey Endelman, and we had all this connection. Also, I wanna say thank you for Intergen. This was our first meeting, 2018, and then this week you guys made an excellent presentation and conference. So with this, thank you all these institutions that I work with. Thank you SDA for the founding. All my colleagues from the potato breeding program here at the University of Wisconsin. Yeah, this is my contact. Thank you.
Thank you, Felipe, for your great lecture. We'll have a few questions now. Can I start it?
Yeah, sure. Go ahead.
Okay. First one.
Thanks, Felipe, for the great explanation. Would weeds increase the noise in identifying the number of plants per plot? Is there any way to separate them from the crop canopy? Yes, the weeds can do that. Thinking on that, we include in our function field crop or field count one parameter called minimum size or mean size, which you can say I only want you counting values above this limit here, this threshold here. Okay, in this case for the weeds, when the weeds is smaller than the actual canopy that we are evaluating. Another way is some index, vegetation index, they can be really related specifically for that crop. For example, I index relate with corn, and then you can make a better mask and remove not only the soil, but also remove the weeds over there. Okay, does that make sense? And also, depending of the tree, the crop that you're working with, there is many machine learning algorithms that you can use. This is a really, really initial possibility, right, that you can use. In our case with potatoes, as you see, as you remember there, we have really nice shapes. We also control the weeds pretty well in the farm. In our breeding size experiment, it was enough using remote sense, actually, image segmentation technique. Okay, thank you for your question.
Okay, I'm going to select another.
Can this text handle with curved rows, no square field trials? That's a great question. Thank you, Danilo. Yes, there is some techniques that you can use. For example, you can make different shape files for the regions in your field that you have a kind of pattern there. So for example, a suggestion you can do first field shape for this part in the field that you have more consistency, and then this other part in the field that has less consistency, you do another field shape, and then you can combine these two different field shapes using one R bind. It's one function in the R. In the tutorial that I talked there is one section called types, I guess types, and I kind of explained this, but the next month I'm gonna present a course for Gen Melhor for the UFV, University of Viçosa, and then I'm going to talk a little bit about that. So contact them to see if they have a spot there. And that presentation is going to be in Portuguese. Okay?
Okay. The next question is from Fernanda Campion. She put a correction after that she means in the image analysis.
Okay. Hello. How do you differentiate low leaf area index, smaller camp, from the stand losses? Thank you for your lecture. That's a really important question. For example, in my case, I'm really using what's important for me is the reflectance for the plant. So we are removing the soil from that point. Okay. If you are interested to know the influence of leaf area index, you should let the soil there. And then you're gonna have, because the leaf area index, is a question. Actually, leaf area index is the proportion of the plant in relation to the area, to the ground. And then for this index, the soil exposed, the presence of soil is gonna push down your scores, and that's important for you figure out this index there. But in my case, I don't care about the soil presence. I only wanna know about my plant reflectance. That's why I'm removing the soil. That's a really great question. Thank you for asking for that. To ask that.
Okay, one other.
Ana Maria. Hi, Felipe. Thanks for the great presentation. Thank you. I'm trying to do my best here. How would you use your program to model heading date, if possible, for small zeros? That's a really great question. Again, I guess if you can figure out an index before, when you're doing the mask to identify only the heading in your crop, you can remove the entire rest. For example, imagine if you have an index only related with green, only green, and the heading there, the inflorescence is brown or red. You can use this index to remove the leaves, maybe remove the soil, and keep only the spots where you have the heading there, and then you can apply your counting. I guess this is one possibility. But actually, this is you should fly really, really close for have enough pixel size to see this, and maybe our software not are able to keep this orthomosaic. Like we have a limitation size over there, right? So you should have smaller size image when you're evaluating in R. What I'm doing, I'm not evaluating flowering time, stage. I'm not evaluating flowers. I'm only evaluating the reflectance. I can reduce my orthomosaic size, and then I run pretty well in R. But sometimes maybe you can also use individual pictures and try a different approach. I don't know if I addressed your question, but it's what I think.
Okay, and the last one, it has two parts. I'm going to put first one and then the other.
Okay, thank you.
This is the first.
Gustavo. Hi, Felipe. In a breeding program, as long as we have a great genetic variability, we can also have a difference in leaf color greenness between genotypes.
Great. The second one.
How do you think this impact the vegetative index use, and how can we overcome that in order to avoid a bias image interpretation? Wait, let me read the first part again.
Okay. Here.
As long as we have a great genetic variability, we can differentially leave. Okay. So if I understood your question, you want to know how the difference during the crop growth cycle can make a difference in your breeding pipeline. The moment that you choose to collect the image should be related with the trait that you want to look for. We're thinking, for example, if we want to evaluate any maturity stage, vine maturity, we need to collect in that point that we are going to collect maturity. So the NDVI in that point is related with the trait that we are seeing directly. Okay, but imagine that I have a disease during the flowering time, and then when I using the same index that I use for maturity, I'm using now for identify yellow regions during the flowering time. And what I have over there, I have a disease, right? So this index is related with that disease. But the best way that you feel comfortable to change the technology, for example, is using the GWAS and seeing if you are select the same region and also repeat the same experiment over time. I don't know if I addressed your question.
Okay, thank you, Felipe, for your lecture and for answers the questions. And so see you really soon, in a few seconds, during the panel discussion.
Okay, thank you.
Oh. So finally, we will enter the panel discussions, but before we start, I'm going to ask you to assess the event feedback form through this QR code there is on the screen, or the link that is in the description of the video. And leave it open so you don't forget to answer it when the live ends. I will give you a few minutes now so you can assess the form. It's really quick and it really helps us to improve our future events. So just to remind you, these are the following topics that are going to be discussed about. And now I will call all the speakers. So welcome again, Felipe, Sara, and Annarita. Are you listening?
Yes.
Yes. So we can start with the first topic, that is prospects for use and challenges of phenotyping.
Okay. Maybe I'm gonna start. Maybe Roberta, I don't know if are you using headphone?
Sorry?
Are you using headphone? I'm listening my voice 2 times. It's okay? You guys can hear me well?
Yeah.
Yeah,
Okay,
we can hear
great.
you.
Yes. I guess when I start here, there is a lot of protocol when you are using drones. You need to have a pilot responsible for the flights, and then in my case, because it was related with my project, I need to took uh, uh, a license for be a pilot, and then it's kind of demanding. And afterwards, each universe has a protocol for fly. In the fields, you need to watch out for people, and what's your protocol in case of accidents. I guess it's super important, but for when you are beginning, and you need to put all these documents together and get all these permissions, is kind of a challenge. I guess it was one challenge that I faced when I start.
Yeah, and I'll add on to that. So when I first started, like I kind of mentioned in my talk, I was kind of just given a drone. I didn't really know what to do with it. I had a friend that had flown a drone before, so he showed me how to like have it take off and land and those little things. But I didn't have any protocols or couldn't find any online on say how high should I fly, what overlap should I use, what do I do with the images afterwards, or really anything that would help me kind of get started. So a lot of it was like trial and error. So one of the biggest challenges for me at first was like so the image GPS coordinates aren't very accurate for most commercial drones. So something I didn't know when I first started is you need something called a ground control point, or something on the ground that you have coordinates for, and then make sure that they're in all of your. You have a few throughout the field. So then, how many do I need? Um, yeah, so figuring out all those little details took me a really long time. So even my first year, the data was pretty much useless. It was mostly to like figure out the best methods to implement. And it's been kind of what I've seen talking to many different students. We all kind of do our own thing, and it's hard to actually like figure out the optimal method. And especially like if you just Google it, you'll have no way of knowing what to do. And there's no online protocols for you to help guide you. So I think that's one of the biggest challenges right now. It's just such a new field, and there's still so much we don't know. And I think we need to do a better job in just getting protocols out there so that other people can use them when they're first starting, and then they don't have to waste a whole year trying to figure things out. And then
Hear you.
Sorry, I have unmuted my computer. I didn't realize it. I think this is something that me and Philippe, we have discussed many times. And the first time at the Phenom, and is that sometimes it's very difficult to find like you can Google something when you are starting a project, and then you don't know the right direction. For example, I think I know Plant CV because my supervisor, she indicated to me, say, oh, because she knew the developers, and then she told me like, oh, you can use it. But at the beginning, I was like searching for a tool, and it was very difficult. So I think the other challenge is that there are very few open source software. Like I was asking you before if IGSoft is open source, because we have another project that we are starting and we need to analyze like something like that. And it's kind of difficult to find open source resources, and not all the laboratory, I think they have the resources to afford some very expensive license. So I think that's another difficult that you can encounter in phenotyping, challenging for high throughput phenotyping.
Yeah, and then I guess that's why, for example, Sara, you and I, we faced this challenge when we were starting. And then one thing that we have in common is like to let all information possible for all people that are going to start and not stay like lost in the world, trying to figure out how to do that, right? So everything that I know, and I know that everything that Sara and Annarita, they know, they put in their papers and pipelines. For example, if you go in the tutorial that I have available, man, what I know is there, right? I don't, everything, if I discover a new thing, I put over there. And I also publish in LinkedIn or social media to help to put this out, saying, people, there is information over there. You are not alone in the world. Because when we start, sometimes we're gonna ask questions, and then we have some problems to understand. Maybe we don't know the language, the software language to write, and maybe you need to start learning a new language to do something that maybe is easy doing another way. That's one thing that we always discuss. For the guys that don't know, actually, Sara and Annarita and I, we met in February in Arizona in a Phenomic conference. Since their time, we are talking about this. We are going forward with our knowledge and trying to get these new people that want to share knowledge. Our idea is
and
always
I
this.
think, yeah, like adding on to what you're saying, so I think it's important not just to share the successes, but when you write a paper, especially in such a field like this one where there's not that much out there, is writing things that didn't work and why they didn't work, how we can improve them. And that way we can, let it all out there, have other people also know, okay, this doesn't work, let's try this other thing, rather than start from scratch and try all these things that we already know are gonna fail. So I think that's one of the most important things, is communicate both the successes and the failures.
I think also like one of the aspects like of phenomic is that having also some kind of agreement on how to get public the data that you obtain, but also like the metadata, which format, which kind of information, because sometimes laboratory they work on the same species also to see if they are doing the right things. It's nice to have some data there that are reliable, they can train their models or things like that. I think so far there is a kind of also confusion about the different approaches, and like everybody is presenting something, but there is not an agreement across all the methodologies. Say, okay, we have to present this data in this way, you have to provide this information, and so people can rely on this. But yeah, so I think that's good also because, for example, when I applied the pipeline, I saw that those some traits, they were not good for rice. So I think another challenge is that many times there are tools that are good for some species, but they are not probably good for another species. And so I think communication between scientists working in phenomics is probably the priority. And I think that's also something positive, because I think in phenomics there is a lot of communication already across science.
That's great. As you saw in my presentation, I said that the way that we are evaluating stand count in potatoes works pretty well because the shape's perfect for that. But when we look at different crop like corn, because the leaves is that way, the algorithm have problem to identify, right? So sometimes works for some crops, sometimes for another crops. And when you have this already established in your pipeline, in your lab, in your breeding program, if you put this available for the public there, would be amazing. I guess, yeah, we should continue using the scientific communication for the future. That's one of these perspectives question there.
Yeah, and I think too, so especially a lot of programs starting up, kind of how what Annarita said, they don't have a lot of resources. So not just sharing our protocols and the software or scripts that we created, but sharing our data as well will become very important. So maybe another researcher at a different university collected similar data to mine, and I can use it as well. And there we have different environments, maybe different species or the same species, different genotypes, and maybe like enhancing that collaboration between different universities will become more important. Yeah, and that goes into metadata. So how we process our data, I think everyone does it differently. How we normalize the data, everything. It's important to document everything with precision, even though, oh, I think everyone does this, I'm not going to even write it down. You should always document every single thing you did with the data, because the raw data and what you actually end up using can be very different.
Yeah, definitely.
Yeah, okay, next question.
Next topic.
Oh, I don't know if we are taking questions, but I think there is Anthony. I don't know if we are doing that, but it's okay.
Yeah,
awesome, Anthony. We also met him in the Arizona conference. We were a really big group there talking about all this stuff that we are talking here right now. That's great.
Do you want me to put the question on the screen?
I don't know. I think it's a good question, because he's asking, are these resources online now? Well, okay, if you want to copy are these resources online now to help people who like to start this kind of measurements for the process? Yeah, I think he was like kind of, hi, Anthony. I think he was kind of saying what we were saying, and that it's something that we already thought at Phenom, I think. So this is why.
Okay,
yeah, from what I've seen, just answering that question, like most papers that I've read don't really have detailed methods or protocols that they use. So that's why I'm assuming my fellows here and I were trying to incorporate that into our papers when we use a method. So I publish all my scripts in my GitHub page and have my protocols there, and also they're highly detailed in the papers. So that's something that we are kind of trying to move towards.
And also, it's not necessarily be a really fancy web page. Just a Word document with some information or directions help a lot.
Okay, can I move to the next topic? Now, entrepreneur. I have a problem with this word.
Sara.
Sara.
Entrepreneurship.
And innovation opportunities in phenomics.
Oh boy.
Sara,
yes.
I'll start. I think for me, I think one of the biggest opportunities right now are in terms of technology development, in terms of both the sensors, platforms, and software that we're using within phenomics. I think in terms of sensors, for example, we want smaller sensors that we can easily put on equipment that's also smaller and cheaper. So for example, a hyperspectral sensor maybe 2 years ago was like this big. Nowadays you can find one like this big, and they just keep getting smaller. So that's one area where there's a lot of maybe opportunities is in sensor development, making sensors that have higher both spectral resolution, spatial resolutions, smaller. And then maybe also making custom sensors, so different wavelengths work for different things. So can we create just a small sensor with the wavelengths that we actually want, measure whatever we are interested in, cheaper as well. And then in software, kind of how we've all been saying, there are very few open source software and even proprietary software. There's maybe a few that dominate each sector. So then having software that's either cheaper or open source also brings a lot of opportunities. So bringing in people from computer science departments, so that's one of I collaborate a lot with the computer science department here. It's how I generated all my code. But they're starting to also get more into collaborating with the plant sciences and other sectors to develop code for us. So that's a realm where there's a lot of opportunities as well.
Yeah, I think like the innovation opportunities, so I think they comes also with the training of people. at the university, for example. So I think also like creating. I remember that at Phenom we saw that the graduate program from I think Iowa University
Amazing.
that were bringing people with different expertise, like in engineering, plant breeding. I think that's a good idea for innovating because I think with training, then you have the best solution for um, to apply in agriculture because you have the expertise of different people, and then you can actually create a solution for the problem that you are looking at. So,
And also with this new technology coming and new companies over there making their own product, the price is going low, and then people are starting being actually it's easier by technology right now. In some countries and some regions, maybe it's more expensive than here in US, but it's there. There's many possibilities how you use these. For example, we three of us here talk about similar protocols applying different ways, different languages and different technology and sensors. So yeah.
mhm. Yeah, I think also one of innovation will be like where to store the data, and resources to analyze because drones and all they are going to produce more and more and more data, and sometimes we need resources like computational resources to manage and handle all this data. So I think innovation needs to go also in this direction.
That's a good point. Actually, we can put this as a challenge from the last
Yeah, it's
question.
true.
The data storage. Yeah, each flight is gigabytes, and each flight that we go to the field is gigabytes.
Yeah,
And
imagine
how
like one of these orthomosaic image that they are so big and sometimes your art crash and like
yeah, and also one of the protocols we need to keep this data for 5 years. Oh boy, imagine that.
I think another area too is with all this new technology. I think a lot of us have been focused on traits that we measure by hand, but we can get so many more traits, and these other traits can also be useful to explain other things. So thinking outside the box and saying, okay, maybe the shape will indicate something about the genotype, or I don't know, something we can use in crop improvement, rather than correlating it, for example, to something very concrete that we measure like height or color or something like that.
yeah.
So just thinking outside the box and coming up with new traits that we can use, I think is also a kind of a exciting new opportunity that this brings.
Yeah, like considering also the talk. So one of the questions that there was me, and also the talk of Philippe that was about the different stage of potato growing. I think also phenomics are providing another dimension, which is like since you take measurements across the time, you are also, if you want to find genes that are activated across the time, you can also say, oh, and I think this is something that with manual measurements, it's very difficult to accomplish because if you think about phenology, you will go, let's say, one time, specific time, but maybe you can do it across time and really like it provides more and more information than manual measurements. So I think that will be for sure an innovation because you can see, especially for very complex traits, you can see which kind of gene are activated across the time while the trait is developing. So that will be something very interesting.
And then I guess here we can talk again about science communication because people we need people doing science communication because the university, the labs, they are generating a lot of information. Some of this information is pretty complex to understand, imagine to apply, right? So when we think the breeder time, when you are there in the field, that guys, we need some easy tools to use, right? Some directly tools to use. It's pretty amazing what the basic science do, but we need these people that are taking the information here and bringing it to us, right? And for we apply this and use in our breeding programs. So this connection, which is science communication, it is super important and also one innovation for the future. I guess any person that decide to work with that will be really successful.
Yeah.
The next question.
We have one question here. Would you like to answer?
Yeah.
Okay.
I'm going to put it on the screen.
So there are so many applications that even include the use of drones to explore Mars. How do you think your knowledge on what you do here on Earth can be applied to the exploration of other planets?
Oh,
I
boy.
think I can briefly answer and then Philippe, you can go.
No.
Okay, go,
I think
Sara.
You can
what
start.
we learn here, what we learn is how to learn something new, really, and how to best practices for gathering data, and also in some degree to think like a computer thing. So we can say, okay, we want to extract this trait, but we have to think, okay, what does a computer see and how it can extract it. And really that can be applied, that has been applied to many different fields. I think one of the major fields is in forestry. So looking at forest species, assessing forest fires, all sorts of things. So I think the knowledge that we gain here can readily be applied really in any field, as long as you know the basic principles of what you're looking at and what you're working with.
You guys remember that that researcher that presented in Arizona, that he was applying research for Mars. So basically, he has
True.
this room where they can simulate the weather and the radiation on Mars, and then he has lettuce, right? Lettuce, and then he were, man, there are people breeding lettuce and vegetables for March already.
Yeah.
And we saw they present in Phenomic 2020 in Arizona this year.
Yeah,
It
it's
was
funny. I
amazing,
remember that.
amazing, super fun. Everything that you imagine, someone in this world they are doing. Yeah.
Yeah. I'm sure that they are already doing it.
It's really interesting. So, do you want to move to the next topic?
Yes.
Okay. This is the final one. Finding a way to enter phenomics.
Well, I think it's kind of challenging. I think it's so it can look challenging, but I think for sure, like so I don't know exactly how you can do it. It's kind of challenging because it's a new developing field. But I think there are now so many tutorials that you can follow. But I think a way to do that will be to contact experts and like get information and resources besides like starting on your own. Because I think it's very difficult. Like let's say, for example, so far they are becoming very popular, the Raspberry Pi, which they can do many things for with like not so much money. But I think it will be appropriate, like if, for example, if I will need to do that, probably I will contact someone that I have read the paper about and ask a suggestion, because it's kind of tricky. So I think probably the best way will be to contact someone that is expert or has done it already. Yeah.
I think when I find kind of getting into phenomics kind of very exciting in general.
Yeah.
I studied agronomy. I've always been kind of into biology and plant sciences. My brothers, on the other hand, and most of my family were all engineers. So I kind of grew up in that world as well. I didn't know anything about it, but I was familiar with it. So when I first started, I just kind of saw it as an opportunity to branch out from the plant sciences and learn about very much different fields. Like I took a computer vision course to learn about computer vision and computer science and how to do some coding. I also met with engineers to figure out how we can adapt a drone, maybe change the sensor, etc., build the hyperspectral system and how we can apply it outside and all those little things. So I love that. I built my own GPS system, so
Oh.
I also got a little bit into like mechanical engineering. I would just call my brother like, "Hey, what do I do here?" So but I like that. I like learning new things, and I think that's something you have to be open to when you get into phenomics. Is you're not just gonna use the data for biological applications.
Yeah.
You're gonna get experience and learn about a lot of different fields.
Yeah, and I guess also it is important, like first, as Annarita said, read a lot, and the main question, I guess, how this technology can make my life easier, or how I can improve my genetic gains. Because if I'm or it's being cheaper to use this technology, less expensive maybe, because if at the end of the day you're not improving that much and you're not having better genetic gains, maybe it's you are using this wrong, or you're not using the right technology. Until you figure out, that takes time. But first, as both of you guys said, ask a lot. Don't feel afraid to ask. Don't feel afraid to contact people and making questions and read. Yeah.
But I think Sara had the good point. I think it's like you have to be open to learn new skills, like to be always like, for example, I come from the genetic world, like, so I have been like for many years like working with genotyping and like genome sequencing and this kind of stuff. So phenomics, it's very new for me, but I think you have to be open to new and learn new skills that are also outside your like education. Like I'm a plant biotechnologist, but so I had to start learning coding and start to learn electronics and this kind of. Okay, for the STEEDS program, probably just Python, and I had already some background. But I think for the new project that I will be involved, and I'm involved, you need to really be open to learn new skills. And I think this is also exciting because you have also many opportunities to kind of evolve, but also like to get your space, you know, because we are early career scientists and we need to also find our independent pathway. And so I think this gives us some new opportunities for development. So, yeah.
I think too, it's such a new field. So, for example, I've been going to Phenom for the past 3 years, and what I saw the first year and what I saw this year are completely different. And the field just keeps evolving each year, and it's just nice to be part of that evolution. So even if you just figured out how to fly the drone and collect data, that's still a lot of progress, and that's still a lot that most people don't know. It's not like you're going into a field that there's already this extensive knowledge and you have to learn all of that before you can actually think about new applications and new discoveries. It's such a new field that it's easy to make your own contribution and be part of that evolution. So I think it's a exciting time to get into phenomics.
Yeah, and I also guess the knowledge is a construction. So something that you use in the past, sometimes when you see a challenge, you can, I can use that. For example, when I started working with remote sensing and potatoes, I started searching for pipelines, and everything was in Python or using expensive softwares. And then I thought, oh boy, I need to start learning Python. And then I started learning Python. But during the process, all the time my brain said, you can do this in our software. You can do this in our software. Because we have this breeding background that use our software a lot, at least where I did my college and my grad school. And then I decided to try, and then works. And then I did the first function, and then the second function. Then we decide put this variable, and because it was working for us, maybe it could work for someone else, and then works. And then I'm here talk about these, and you guys, the same way you guys are here because you also use some knowledge that you had adapting this for your challenge, and you guys share that, right?
Yeah. Yes, totally agree. Yeah, I think there are some questions.
Okay, we have one question. I'm going to put it on the screen.
What do you think a successful phenomic program at university should include in terms of classes? Now that you are world experts, what would you suggest to us at the university level? Oh, that's a good question. Well, I think for me, it's kind of so I started phenomics when I was already in my post, but I think it's something that should absolutely be included at university level. And I think it's so. I think that some of the skills that they need to be taught at the university is for sure coding. So that's for sure basic like R or Python or Unix. Maybe some robotics if you are going to use like drone. But also I think concept about image segmentation, like the different color space, what you need to do when you have image, and because that is very important from very simple project like could be the seeds to very complicated like drone images and hyperspectral data. So I think for me, but maybe there are more things to include. I don't know if you, Sarah, do you have phenomic class at your university? Because
So actually, even in my PhD program, there aren't any currently. I actually just I was in contact with a computer science professor, so I took his course, and there's a lot in the kind of we got the natural resources department, not in agriculture, but those are more again for like forestry applications, and those actually have some remote sensing classes, not directly phenotyping, but it gets into the idea of remote sensing, so how to collect data remotely using sensing technology like cameras. So those type of classes exist. I think they just started both undergrad and graduate course on phenomics this past year. It's the first time. So it's something that's kind of starting to get implemented in curriculum, but it wasn't available when I was taking courses, unfortunately.
Yeah,
sorry.
Yeah, go ahead.
Something I think is very important too is just like how to deal with big data. And you learn that even in genomics courses, but how you have all this data set, how to kind of mine the useful parts, get rid of noise through normalization techniques, and how to basically just handle large data sets, maybe through different coding programs like R, Python, but then also where to store them, how to kind of draw them back, and so all those little things are important.
Yeah, that's our really important classes. Yeah, back in the university where I did my doctorate degree, University of São Paulo, ESALC, USP, we did have different departments, and some departments more specialized for remote sensing and other for data analysis, but all classes, it's open, then the students can take these classes. The breeding, back in my time, the genetic department has a course for phenomics, and then I did, it was amazing. I could learn a lot. So yeah, we do have this class in my genetic and plant breeding department there. But I guess it's something that is coming really strong now. Some people can start to contact me and asking if could use, for example, our pipelines that we are put over there in their classes, and I guess yes, that's why we are putting there for you can use download the example. Also, is important put your examples for people to download and follow the steps. But yes, we do have.
But yeah, I think for the future of like agriculture school, phenomics needs to be a must. Like, needs to be introduced at all levels, from undergrad to PhD school. Yeah.
Yeah. Any other question?
So, is it finished?
Yeah, probably me and Philippe, we want to announce that, like, since these talks, like what we say is this panel discussion is that we need to have open resources and training and to put together people. So we are going to organize a series of workshops in this fall, online for free. So keep like how to say, like, keep connected, be connected.
Yeah, keep following
Yeah.
our social media, and as soon as possible we're gonna start announcing our webinars, and we are trying to get really amazing people. Everything is open source, so think on that. Okay, just keep
Yeah.
following us.
Yeah, they
Okay.
will be like for free and
Yes.
like open source data, and there are very good speakers in different fields, like different kind of application of phenomics.
Awesome.
Okay, when you have more information, you can send us the flyer, and we can help you to promote the event.
Great.
Great.
Thank you.
So finally, we achieved the end of this live. So on behalf of Jivank, I would like to thank you once more for.
Thank you for
Thank
inviting
you
me.
for the invitation.
Yeah.
Thank you, guys. You're doing a really great job. Yeah, thank you.
Yeah, very good.
Yeah, we are really happy with the result of the live, and I'm sure that everyone who watched it has enjoyed a lot too. So thank you all the followers that have watched the live, and we hope to do more events like this in the future. So keep in touch.
Please do.
So
Bye
that's
bye.
it, guys.
Well,
Bye bye.
thank you.
Bye.
Bye.
Thank you for listening.
Bye. Bye.